Alright, here's a short video on synapses. Right, so we've got, just as a refresher, I'll draw the standard multipolar neuron. Remember, they certainly don't all look like this, but we've got our standard multipolar neuron. Branching at the end of the axon, right? So soma and dendrites. Super important axon initial segment where the cell integrates the input at the soma and dendrites, right? Stimulatory and or inhibitory input to determine whether or not we reach threshold of voltage gated channels, right? If we do, then we get an action potential. Well, we get an action potential, and then we would get repeated action potentials, which is an impulse, nerve impulse. And yeah, it can be either continuous propagation if it's unmyelinated, or it can be saltatory propagation, which is myelinated, which means the action potentials only occur at nodes. So I can actually draw some myelin on here if you'd like. Those are supposed to be cells wrapped around the axon. And of course, the action potentials in that case can only happen at the nodes in between those myelinated regions. Why do we need the nodes? Well, remember, electrotonic current still decays even when we have myelin, and so we have to boost the voltage change, right, at the node by allowing more sodium influx, right? That's the initial influx for the ion current during an action potential. And so we just have these spots where we boost the signal essentially, and then it travels passively, and then we boost it, then it travels passively, then we boost it, all the way to the end of the axon. And again, if you had an axon that ran all the way to the sun, an action potential started all the way down here on Earth would travel all the way to the sun, right? It's not, it travels to the end of the axon because it keeps getting regenerated along the way, right? Boosted by the voltage-gated channels opening and changing the voltage of the cell again to open more voltage-gated channels. So it's a positive feedback loop, right? Okay. At the end of the axon, we have potentially, usually, telodendria, the branches, to form many synaptic terminals or synaptic knobs on the target cells. So our target cells are out here. Right? And so now what we want to do is we want to focus on what happens at the synapse and how this cell tells these cells how to change their function. Right? Now, before we do that, we have to talk about the fact that there are actually more than one type of synapse. We have the two main categories are chemical synapses, and the other version is an electrical synapse. Chemical synapses are much more common. And they are synapses that use a chemical. In other words, so these are synapses that release a neurotransmitter. They can fall into or be broken up into essentially two categories. Ionotropic channels, ionotropic chemical channels, and metabotropic. Now, what we've sort of focused on so far when we brought it up and we talked about input at this end of the cell, right? We talked about EPSPs and IPSPs. We were talking about ionotropic chemical channels. In other words, a neurotransmitter affecting ions, right? Ion movement across the membrane, right? So these are ligand-gated channels that cause the channels either open or sometimes even close, but usually open to allow ions to move to change the membrane potential. So the ionotropic ones form those groups. EPSP generating channels and IPSP generating channels. Oops. Right. Okay, hold off on that. We're going to come back to those. Again, that's the most common type. Metabotropic, they're not opening channels, at least not directly. And so what they're doing is they're changing the function of this cell but not doing it through ion flux. And so they're linked to what's called a second messenger system. So the chemical will bind to, the neurotransmitter will bind to a receptor. And what that usually does is it changes the activity of enzymes in this postsynaptic cell to alter its function. So it's changing the metabolism, if you will, of that cell, metabotropic synapse, right? And so those tend to have longer acting effects. The ionotropic, right? You can open and close ion channels very quickly. The pumps and channels, the resting memory and potential factors will put that ion right back where it was, and the effect is dissipated. Metabotropic tend to last longer because we're tending to change the activity of cellular proteins through the reaction, and those tend not to go on and off as quickly. So slower action onset, and they tend to last longer. Okay. Metabotropic is just an example. Metabotropic receptors are involved in changing the connectivity between cells, and that's really important for things like memory. In memory, we want to change the connectivity here, and what that means is we actually want to change the number of receptors in the membranes of these cells so that they'll respond more or less to a particular neurotransmitter, right? And that's actually one of the way memories are formed is by enhancing the connectivity between neurons through that sort of changes of the memory proteins that connect the cells, right? These are the proteins involved in conveying the signals from cell to cell. Electrical synapses are really easy. They're also very rare. They tend to be in the central nervous system. A lot of invertebrates have them, even outside of their central nervous systems. And these are synapses between neurons formed by gap junctions. So the interesting thing here, let me go backwards, right? When we talk about this cell and this cell, that's a unidirectional flow of information. This cell releases neurotransmitter onto this cell, right, to change its function. There's no information going backwards, okay? Or there can be, but it's not the norm, okay? With gap junctions, right? Gap junctions, we have two cells that sort of butt up against one another. We'll just have one gap junction here. So here's where these two cells sort of meet, right? And they each have a pore that docks. And when the two pores dock, you have the formation of a complete gap junction. And what can happen is ions can move in either direction across that gap junction. So this cell depolarizes, electrotonic current will travel to that cell, and if that cell depolarizes, electrotonic current would travel to this cell. Right? And so these are bidirectional synapses. They're super fast because there's no chemical released. It's electrotonic current. The traveling of electrotonic current is blazingly fast. And so where do we find electrical synapses? Well, where we need things to be really fast. Invertebrates actually employ them for that reason, adaptive purpose. In other words, you can very rapidly stimulate the swim muscles in a crayfish, for instance. Okay? By having gap junctions, taking the slowest component out, the slowest component being the synaptic events at chemical synapses. Take that out and just have current flow between the cells, and things happen very fast. The other adaptive purpose of this type of synapse is they allow cells to be synchronized in their function. And so there are CNS neurons that are stimulated in groups, and you get this sort of like cyclic patterns of stimulation. Consciousness is thought to be maintained by this sort of cyclic pattern of activity along circuits of neurons connected by gap junctions. Okay, so anyway, coordinating multiple cells to work synchronously is another key function of gap junctions. Now let's go back to the ionotropic, again, because that's probably the largest, most common group that people talk about. Not that the others aren't important, but these guys are found in more places. And so let's talk about how an ionotropic synapse functions. All right, now this could be another neuron. I'm just drawing it as a generic cell here. Could be another neuron though. This is our presynaptic neurons, right? This is our synaptic knob, synaptic bulb, and in the synaptic knob we have synaptic vesicles, if you will, neurotransmitter containing packets, right? Little membrane-bound bags of neurotransmitter, sort of sitting there waiting to be stimulated, to be released. And these are actually produced upstream, and then they're conveyed down the axon along a microtubule system. There's something called axoplasmic transport, where these vesicles are marched down the cytoskeleton by what are called motor proteins. Right? It's kind of crazy cool. It's like a little conveyor belt that carries these vesicles down. I mean, this axons can be very long, can be feet long, right? And so you have to march these vesicles continuously down to replenish them at this end of the cell. Now there are millions of vesicles here, right? And each vesicle contains thousands of molecules of neurotransmitter. And so I won't add it to all, but remember that each of these is going to have lots and lots of neurotransmitter molecules. So when an action potential, a nerve impulse, but an action potential is generated here, right? We're going to depolarize the membrane of the synaptic bulb. What we need to have happen is we need to have exocytosis. Now, they're not all going to exocytose at once. There's a store there. We're only going to stimulate the exocytosis of a small group of them, right? And so when we depolarize due to an action potential arriving, right? Remember that depolarization would be due to sodium channels, right? Voltage-gated sodium channels. But when we get to, and I'm going to color code this, when we get to this membrane, right, things change a little bit. We still have voltage-gated potassium channels, and we still have voltage-gated sodium channels, but we also have voltage-gated calcium channels. And so what we're going to get is calcium influx. And we know it's going to be calcium influx, right? So this is a channel. It's a gated channel, so I should put a door on it here, right? We know that calcium is going to enter because the inside of the cell is negative before it depolarizes, right? Electrical gradient inward, and the chemical gradient is inward. So in plus in equals in, right? If you recall, so the equilibrium potential for calcium is like plus 124, so it's hugely positive, and so we know that's going to come in. Calcium is the stimulus for the vesicles, some, not all, a small number of these vesicles to fuse with this membrane. So I'm going to make a little spot here where our vesicle, one of our vesicles at least, has fused with that membrane because an action potential has arrived and allowed calcium influx into the synaptic bulb. And we're going to release the neurotransmitter, a little bit bigger so we can see them, diffuse into the synaptic cleft. Right? So this cleft is the space. Now that neurotransmitter is going to diffuse anywhere and everywhere, but one of the things it's going to do is diffuse across to this postsynaptic cell. And as long as this guy has a receptor for it, we're going to get, remember we're talking about ionotropic synapses here, and so we're going to have a receptor on a channel. And so I'm going to put a little pocket on this guy, right? It's got a gate on it, and I'm going to show the gate is already open because I'm going to have a neurotransmitter in that receptor site. Right? So that is a ligand-gated channel or chemically gated channel, right? The ligand being the neurotransmitter, right? That's the neurotransmitter. There's a variety of neurotransmitters. When you do your reading, you'll see acetylcholine is a very common neurotransmitter. Norepinephrine is a neurotransmitter. Dopamine, GABA, serotonin, on and on and on. There's a whole variety of them. And many of them are stimulatory. Some of them can actually be inhibitory to the actions at this cell, right? They could cause channels to close, or they could have, if they're metabotropic, they could inhibit the cell through metabotropic responses. But now we have to decide, well, what kind of channel is this, right? And again, that would be specific to this particular synapse. If this is a sodium, ligand-gated sodium channel, right, then sodium from the extracellular fluid would enter the cell. And you would end up with an EPSP, right? It's an excitatory. It's stimulating the cell, right? If, on the other hand, it's a ligand-gated potassium channel, and actually the same would be true if this was a calcium channel, right? EPSP. If it is a ligand-gated potassium channel, then potassium would leave the cell, very negative equilibrium potential, minus 95, and you'd be left with that picture, right? And remember, in either scenario, lots of similar charge do not want to hang out in the same region. The rest of the cell is, in this scenario, more positive than this particular region, and so you get electrotonic current outward. Right? Dissipates with distance, and again, if this is a neuron, this would be inhibitory to it because it's pushing you away from the threshold of the voltage-gated channels. If this were, go backwards, if it was sodium or calcium, then these would be plus signs, and that would be stimulatory because it's pushing you toward the threshold. Remember, the threshold is more positive than the resting membrane potential, right? Resting membrane potential in neurons about minus 70, threshold about minus 65. So if we become more positive, we're excitatory because we're getting closer to threshold. If we're becoming more negative, right, this would be an IPSP because it's tending to push us away from that threshold and away from generating an action potential. Okay. And again, if it's not a neuron, this target cell, then we could just be the depolarization or hyperpolarization, right, could be stimulating or inhibiting the actions of this cell. This could be a muscle cell, right, not a neuron, in which case we could either be stimulating or inhibiting its contraction. Okay, so that is an ionotropic synapse. Now, one of the things about ionotropic synapses is that we can get more than one active at a given moment. So I'm going to add another cell. And we've seen this before, right? When I first drew a neuron and I showed synapses with it, I showed you with more than one synapse, right? And so I could have another cell sort of coming in here, releasing its own. Now there could be more than one synapse from a given cell, from a single cell. I'm drawing this as two separate neurons having input. And so in this scenario, they could be fighting over control here, right? Now they could both be, right? Let's have this one releasing some neurotransmitter. And we'd have to have channels. Right? If they were both ligand-gated sodium channels, well, or potassium channels, whichever we've got here, then they would be working together, right? They would be tending to push this cell in the same direction, right? They could be pushing it in the opposite direction. In other words, if this was a ligand-gated sodium channel, it would look like this. With my electrotonic current arrow there. Right? And then we have that scenario, where one cell is being stimulatory, EPSP, and the other cell is being inhibitory, IPSP. And they would add together, right? And again, if it's a neuron, then at the axon initial segment, it would depend on which one was bigger, essentially, right? As to which way it's going to be. Is it going to be stimulatory overall, or is it going to be inhibitory overall? That action is called summation, right? It is one of the qualities, right, key qualities of graded potentials. They can be different sizes. I can open one channel here and 3 channels over here, right? Graded, they can be in different sizes. They dissipate with distance. They are not propagated, right? This is electrotonic current, not propagated like an action potential. They can be stimulatory or inhibitory, right? But they also can summate. They can be added together. They can be added together over time, or they can be added together spatially. So in this scenario, we're adding two together. Like if these both happen at the same instant, they would be summating over space on this cell, which is called spatial summation. Let's squeeze that in up here. A really good example of spatial summation is controlled respiratory muscles. Right? So your diaphragm, for instance, is controlled by a multipolar motor neuron. Imagine this guy, I'm going to put another one up here, had two, and it's not going to be two, but for simplistic reasons, had two synapses on it. One is from your respiratory centers that is input from your oxygen levels and carbon dioxide levels that basically says contract, breathe, contract, breathe, contract, breathe. Right? So let's say that's this one. It's causing an EPSP, we get an output, we get contraction of the muscle downstream here. Can you stop breathing right now? Can you hold your breath? Of course you can. That doesn't mean that this input from your respiratory centers stopped arriving. It just means that there's another input, right? Your conscious input through a separate pathway that basically says, ignore that and hold your breath for a minute, right? And so, right, now we've got this battle going on where one's generating EPSPs, the other's generating IPSPs, and as long as you continue to hold your breath, the IPSPs are winning, right? This one would be winning over so this would be conscious control in this scenario. This would be unconscious control, right? This is your respiratory center saying, breathe, dummy, breathe, right? This is your conscious control saying, no, we're going to hold our breath for like 2 whole minutes. Well, the oxygen levels are falling and falling, and the input along this pathway gets bigger and bigger, the unconscious pathway, right? And it becomes more and more stimulatory, right? It gets harder and harder to hold your breath. If you had really strong willpower, you could hold your breath until you passed out. Well, guess what happens to the inhibitory input, the conscious input? Poof, gone. Who's left? Right? The stimulatory input. You start breathing again. Right? So a little kid that says, I'm going to hold my breath until you give me, some candy or whatever. Say, go for it. Right? They might pass out, and then they'll start breathing again. Right? So again, but that's a good example of spatial summation, right? Where we're adding together, in this scenario, EPSPs and IPSPs. It can be all EPSPs or it could be all IPSPs that summate. Another version is, let's forget about this guy for a second, and imagine this guy has action potential after action potential arriving. And what's going to happen is we're going to get a packet of neurotransmitter and open channels, and another packet and open more channels, and another packet and open more channels. And so what's going to happen is this is going to get bigger and bigger and bigger. And again, the local current would get bigger and bigger. That is summation that's occurring over time because each action potential is occurring slightly after the previous one. That's called, and so in time, the term you use is temporal. Right? Can you get temporal summation and spatial summation at the same time? Yep. Right? You get one fire that's firing repeatedly, but if you add another one anywhere else, you're also getting spatial summation at the same time. Okay. And that's all added up by this guy to determine, right? Remember that postsynaptic cell is integrating the input to determine what it's going to do in response. Okay, so our chemicals, to summarize, our chemical synapses are ionotropic chemical synapses. Utilize a neurotransmitter. They open ion channels. They're relatively slow compared to the electrical channels. The slowest part in neural transmission, right, is that part. That's diffusion across the cleft. Electrotonic current down the axon is blazingly fast. This synaptic transmission part, the vesicle releasing the neurotransmitter and it's diffusing and the binding receptor, that's called synaptic delay. It's the slowest part. What it means is pathways that have lots of synapses in them, called polysynaptic pathways, tend to be slower than synapses neural pathways with fewer synapses. The least being what's called a monosynaptic reflex, where there's only one synapse between a sensory neuron and a motor neuron. It happens right in the spinal cord. Okay, so there is this other term and potential for what's called synaptic fatigue. If this guy fires, again, when we talked about refractory periods and how short they are, you can get hundreds and hundreds of action potentials per second. Well, if that were to go on for minutes or even hours, you could run out of neurotransmitter here, right? Or at least it would be hard to resupply at a rate that would keep up with neurotransmitter release. In which case, your effect on the postsynaptic cell would sort of diminish with time. That's called synaptic fatigue. It's pretty rare, but it can happen, and it's due to limitations of this chemical messaging between the pre and the postsynaptic cell. Okay, one other version of synapse I wanted to talk about, because so far we've talked about, again, when it's neuron to neuron, we've talked about axodendritic synapses, like this guy, axon to dendrite. We could talk about axosomal synapses, which really aren't much different, but they're on the soma, like so, right, instead of on a dendrite. Okay, there's another version called axonal. And what these guys do is they synapse, figure out where I want to add them here. They do not synapse here. They synapse way over here. Right, so I'm going to put them on here. Like way at the very end of the system. These are like, I call them like sneaky synapses. They're not fighting up here over whether or not this guy generates action potentials or not. What they're doing is saying, all right, I'm not fighting that battle. What I'm going to do is I'm going to adjust the depolarization here to adjust the amount of neurotransmitter that's released. So they're sneaky. They're not saying, okay, well, I'm going to try to fight whether it generates action potentials or not, but I'm going to enhance or inhibit. So it's called presynaptic facilitation, enhancing, or presynaptic, right? This is before the synapse. Presynaptic facilitation, increasing the output here, or presynaptic inhibition, decreasing it, right? And guess what? We can talk about, the really easy answer is if you depolarize less, in other words, if we either limit the calcium influx here, right, or minimize the size of the action potential through ion currents here, right, add more potassium flux, for instance. Potassium would tend to keep it from becoming so positive, would minimize the calcium channel opening, then we release less neurotransmitter. If this guy opened calcium channels directly, then we'd add more calcium, we'd get more neurotransmitter. So think of axonal synapses as not fighting for control. They're not doing summation. What they're doing is turning the volume up or the volume down on the output of that neuron, right? Assuming that, there'll be some sort of output here. It's just going to adjust that output. Again, that's an axonal synapse. Last thing I'll say is a little bit about what are called neuromodulators. Neuromodulators are often released by neurons, but they can be released by other cell types as well. Remember the glial cells, they can release neuromodulators. Endorphins are a form of neuromodulator. There are also exogenous neuromodulators like caffeine. These are molecules that affect the sensitivity and action of neurons and the target cells, right? And so they tend to have what are called, again, those metabotropic effects. They're tending to sort of adjust the long-term functioning of these cells. They can act on the presynaptic cell to adjust the output of neurotransmitter, or they can affect the postsynaptic cell by adjusting the sensitivity to the neurotransmitter that's released. I mentioned the endorphins, right? They tend to modulate pain, right? Heard of the runner's high, right? When you exercise, you release endorphins and enkephalins, similar molecule. And what they do is they tend to inhibit pain pathways by adjusting the neuronal connectivity of those pathways, right? Again, tend to be metabotropic and more longer term acting than a neurotransmitter. Now, longer term is relative. Neurotransmitters have half-lives that are really short, right? You can contract the muscle and then not contract it in a split second, right? Whereas neuromodulators would tend to have half-lives that are probably more along the lines of minutes, and maybe even longer than that, but they're tending to have effects that last a little bit longer than neurotransmitters. All right, we got through chemical synapses, electrical synapses, and axonal synapses, and with the ionotropic, right? IPSP, EPSP, we did the summation, right? Another really important characteristic of synaptic function is the idea that you can add together graded potentials, right? Either in space, spatial summation, or over time at a given location, at a given synapse, right? That's temporal summation. All right, have at it.